Imagine for a second that you have, like, a coworker. You've known them for a long time, you sit near each other, you chat by the coffee machine. The relationship is completely neutral, maybe even pleasant.
Right, just a normal, everyday dynamic.
Exactly. But one day, out of absolutely nowhere, they just snap. They start screaming at you, totally unprovoked. And the thing is, your memory of who they are hasn't changed.
Yeah, you still know their name, what they look like.
Right where their desk is.
Right.
But your feeling about them, your visceral emotional response is completely flipped. You now avoid them in the hallway at all costs. So the question is, how exactly does the brain rewrite that emotional tag without erasing the person's core identity?
Well, it's one of the most fundamental questions of survival, really. What you're describing is a biological concept we call social valence.
Social valence.
Right. It's the positive or negative emotional weight that your brain assigns to a specific individual, purely based on your past interactions. And the ability to continuously and rapidly update that valence is absolutely crucial.
Because things change, right? People change.
Exactly. It allows social animals and, including humans, to navigate highly dynamic social hierarchies. You have to know who is safe, who is a friend, and who has suddenly become a threat. And you have to do all of that while minimizing the energy and danger of constant conflict.
Which brings us to today's deep dive. We are centering on a brilliant piece of recent research that literally traces this process cell by cell.
Yeah, it's a fascinating study.
We are looking at a 2026 paper published in the journal Science by a team of researchers, Sudo and colleagues, titled Neural Circuits for Valence Updating in Social Memory. Our mission today is to meticulously unpack the exact neural machinery behind this phenomenon.
And we're going really deep here.
Oh yeah, we aren't just going to tell you what happens. We're going down to the microscopic level, from the memory center to the fear center, all the way down to the action center.
And I think the reason this research is so groundbreaking isn't just because of their theory. It's because of the sheer precision of the tools they used to prove it. I mean, they didn't just observe mice fighting and make assumptions.
Right, they actually got in there.
They physically manipulated the neural circuits in real time to isolate the exact synapse where an emotional update happens.
So let's start with their setup, because I found the behavioral algorithm they tested to be just fascinating. To figure out how we update fear about a specific person, you can't just wait around for mice to naturally get into a fight in a cage.
No, you'd be waiting forever and it wouldn't be controlled.
Right, you have to manufacture the conflict perfectly.
Yeah.
So the researchers took a subject, mouse, our main character here, and familiarized it with two neutral mice. Let's call them mouse A and mouse N.
And they left them together for a while, right?
Yeah, for 72 hours. Just hanging out, getting to know each other, building a memory.
Right, so they were establishing a solid baseline of familiarity. But then comes the manipulation. To see how the memory updates, the researchers needed to turn mouse A into a vicious aggressor on demand.
While leaving mouse N completely neutral.
Exactly. And to do this without traumatizing mouse A or using, external shocks, they used a mind-bending technique called chemogenetics.
Wait, chemogenetics? I've heard of optogenetics, where scientists use light to turn brain cells on and off. But how do they use a chemical to target only the aggression cells? Are they just injecting a drug and hoping it goes to the right place?
That's the elegant part of this tool. It's a two-part lock and key system. So the researchers targeted a very specific tiny region deep in the brain of mouse A called the ventromedial hypothalamus.
The ventromedial hypothalamus.
Yeah, or VMHVL for short. We know from decades of research that this area is the command center for aggressive territorial behavior, but they didn't want to stimulate the whole region.
Because that would just make the mouse a generic monster, right?
Exactly. They specifically hunted down a subpopulation of neurons in this area that express receptors for the hormone progesterone.
Okay, so they found the exact cellular zip code for aggression.
Yes, exactly.
Yeah.
Then they used a harmless viral vector. They essentially hollowed out a virus, packed it with a custom genetic code, and injected it into that specific zip code.
Wow, okay.
And the virus infected those exact progesterone-expressing neurons and forced them to grow a brand new synthetic receptor on their surface, a custom lock that doesn't exist anywhere else in nature.
So mouse A now has this custom lock on its aggression switch. What's the key?
The key is a highly specific designer drug called DCZ. On its own, this drug does absolutely nothing to a normal mouse. But when they injected mouse A with it, the drug circulated through the blood, found those synthetic locks in the hypothalamus, and blasted those specific neurons wide open.
It artificially induced intense, uncontrollable aggression.
Right. Mouse A violently attacked and defeated our subject mouse. Meanwhile, mouse N, who didn't get the designer luck or the drug, just sat there peacefully.
So our subject mouse just experienced a random severe trauma at the hands of their old buddy, mouse A. And the key finding is how the subject mouse processes this. Afterwards, the subject mouse avoided only mouse A.
Yeah, it was highly specific.
They stayed away from the aggressor, but were totally fine hanging out with the neutral mouse N. The researchers call this specific social avoidance.
Right.
But here is where the clinical implications just blew my mind. If the researchers skipped that initial 72-hour get-to-know-you phase and just had a total stranger, mouse A, attack the subject mouse ended up avoiding both mouse A and the innocent bystander mouse N equally.
Yes, and if we connect this to human psychology, it's profound. The attack from a complete stranger mimics the mechanism behind generalized anxiety disorder, or broad social withdrawal.
Really?
Yeah. Without a pre-existing memory of the individual, the brain's algorithm defaults to a defensive generalization. It basically says, I was attacked in this environment by a mouse, therefore all mice in this environment are dangerous.
It's like if a stranger yells at you in a coffee shop. You might be so shaken up that your brain flags the whole location as a threat.
Right, and you just avoid that coffee shop entirely.
But if your best friend suddenly yells at you in that same coffee shop, you just avoid your friend. You don't avoid the innocent barista.
That is a perfect real-world translation. The performance memory acts as a biological anchor. It allows the brain to restrict the newly acquired negative valence that fear tags specifically to the individual who caused the harm,
thus preventing a catastrophic generalization of fear to the entire social world.
Exactly.
Okay, so if you are listening and mapping this out in your head, let me pause and synthesize this first crucial takeaway. Let's do our first bulleted recap of the deep dive.
Good idea.
Bullet 1. Social valence is the emotional tag we put on specific people. And bullet 2. Prior familiarity, having a memory of someone, is actually a biological shield. It's absolutely required to attach negative valence to a specific individual rather than generalizing fear to everyone.
Spot on.
So a question for you, the listener, as we go deeper. If prior memory is the shield, how does the brain actually store that specific identity while simultaneously updating the fear? Where's the file cabinet?
Well, to answer that, we have to look at the brain's physical wiring. The researchers trace this behavior down to two distinct neural highways that originate from the brain's memory center.
Okay, break that down for us.
Sure. So the identity of the specific mouse, what neuroscientists call the engram, or the actual physical network of cells that hold a memory, is stored in a region of the hippocampus called the ventral CA1. Let's just call it the VCA1.
Right, the hippocampus is the classic memory filing cabinet. But from the VCA1, the road forks, right? Why, two paths. Doesn't the memory just get rewritten in the hippocampus and then go straight to the fear center?
You'd think so, but no, the brain separates the data streams. The VCA1 sends physical projections axons down two completely different pathways.
Where do they go?
One highway goes directly to a region called the nucleus accumbens, or NAC. This area is famous as the brain's reward and physical action center.
Okay, action center, got it.
The other highway takes an indirect route, projecting from the memory center to the basolateral amygdala, or BLA.
And the amygdala is the fear center.
Exactly. It's the primary emotion and fear center.
So we have a memory to action highway and a memory to fear highway. And to figure out which one was doing the actual valence updating, the researchers used optogenetics. Now, you mentioned earlier that chemogenetics uses drugs. Optogenetics uses light, right?
Right. Exactly.
How does shining a light inside a brain tell us what a circuit does?
Well, instead of a custom lock that opens with a drug, optogenetics uses a custom lock that responds to specific colors of light. They used another harmless virus to insert a light-sensitive protein into the VCA1 memory neurons.
Okay.
This specific protein acts as a molecular gate. When scientists shine a yellow laser light onto the brain through a tiny fiber optic cable, that protein forces the neuron to shut down completely.
So it basically sets up a physical roadblock on the highway.
Yes, stopping any electrical signals from passing through.
That is wild. They can just flip a light switch and silence a specific memory pathway. And the results here isolate the mechanism beautifully. When they shine the yellow light and block the pathway from the memory center to the action center, the VCA1 to NAC root, the mouse still avoided the aggressor.
Right, the fear was totally intact.
But, and this is the crazy part, when they move the roadblock and block the pathway from the memory center to the fear center, the VCA1 to BLA route, the specific avoidance completely vanished. The mouse just stopped avoiding the aggressor.
Which tells us exactly how the labor is divided. The direct circuit to the action center handles the familiarity, the raw recognition of the individual's identity. But the circuit to the fear center, that indirect route through the amygdala, is the one solely responsible for actively updating and holding the emotional valence.
So the memory of who this is and the memory of why I should be scared of them are traveling on two completely different physical cables in the brain.
Precisely. And this separation is a vital feature, not a bug. The brain separates the raw identity from the emotional weight.
That makes so much sense.
That is the exact mechanism that allows you to remember the coworker who yelled at you. You still know their face and their name without losing the concept of who they are. The VCA1 holds the identity file, but it has to constantly talk to the fear center to update the emotional warning label on that file.
Okay, let me pause again to make sure our mental map is crystal clear. We've got our second big takeaway here. The VCA1 is the memory center. It sends the raw identity down one path, but it uses a very specific highway to the basolateral amygdala, the BLE fear center, to update the emotional tag.
Exactly.
But this brings up a huge question for me and hopefully for you listening. We know the highway now, but what is actually happening at the destination? If I drive down the highway from the memory center to the fear center, how does the message actually get rewritten at the cellular level?
Well, to see that, we have to zoom in even closer. We need to look down at the microscopic level, right at the synapse.
The synapse, meaning the gap between the cells.
Right, the tiny gap where the axon from the memory cell connects to the dendrite of the fear cell. The researchers found that the memory cells from the VCA1 connect to a very specific subpopulation of cells in the BLA. These are dedicated fear neurons that express a unique genetic marker called ARSPO2.
ARSPO2. Okay, I'll just picture them as the alarm bell cell. So what physically happens at the synapse between the memory cell and this alarm bell cell when the mouse gets attacked?
The physical connection gets stronger. This is the fundamental biological basis of learning. After the social defeat, the researchers took acute brain slices and measured the electrical properties of these specific synapses.
And what did they find?
They found a massive shift in the types of receptors sitting on the surface of the fear cell. Specifically, they saw a significantly increased ratio of AMPA receptors compared to NMDA receptors.
Okay, I remember AMPA and NMDA from like a college biology class. The receptors for glutamate, the brain's main excitatory chemical.
Yeah.
But how does changing the ratio actually update a memory? What's the mechanism there?
It's one of the most beautiful mechanisms in biology. Think of NMDA receptors as coincidence detectors.
Coincidence detectors.
Yeah. Normally, they are plugged up by a magnesium ion. They only pop their plug and open when two things happen at the exact same time. The memory cell fires. AN, the fear cell, receives a massive shock or trauma signal.
Okay, so they need both.
Right. When both happen simultaneously during the attack, the NMDA receptors open up and let a flood of calcium into the fear cell. This calcium surge acts like an internal construction crew. It triggers a chemical cascade that physically drags more AMPA receptors from inside the cell up to the surface of the synapse.
And AMPA receptors are the ones that actually catch the routine electrical signals day to day. So
Yeah.
I've always thought of this like a volume knob.
Yeah.
By putting more AMPA receptors on the surface, you are just turning up the gain on that specific connection. The attack physically turned the volume of the mouse, a signal way, way up inside the fear center.
Yeah, it's close to a volume knob, but biologically it's more like adding extra speakers to a sound system. By pulling more AMPA receptors to the surface, the physical surface area for receiving the signal actually increases. The connection is wider, denser.
I see.
So now even a tiny whisper from the memory cell, just the mere sight of mouse A's, gets caught by all these new receptors and blasts a huge deafening alarm signal into the amygdala. This process is called long-term potentiation, or LTP.
That is incredible. But here's the part of the paper that genuinely sounds like science fiction to me. If that specific addition of AMPA receptors, those extra speakers, is the literal physical manifestation of the fear, the researchers theorized they could erase the fear by removing the speakers.
Yes.
And they actually did it using something called optical LTD.
Yes. Long-term depression. It's the biological reverse of learning. They had already tagged the exact memory cells for the aggressor mouse with a blue light-sensitive protein. This allowed them to artificially activate just the memory of the attacker.
So they had the specific memory cells wired to respond to a blue laser. What did they do with it?
They flashed the blue laser at the connection in the fear center at a very specific slow frequency, exactly 1 hertz or 1 pulse per second. In neuroscience, this slow, rhythmic stimulation mimics the brain's resting or forgetting states.
Okay, so it tricks the brain.
Exactly. It causes the synapse to do the opposite of what it did during the trauma. It actively swallows those extra AMPA receptors back inside the cell. It takes the extra speakers away.
They literally reached in with light and downgraded the connection.
Yeah.
And the behavioral result.
The mouse completely stopped avoiding the aggressor.
Wow.
The identity memory was intact. They still knew it was mouse-eyed, but the specific fear was physically erased from the synapse.
It is a stunning demonstration of causality.
Yeah.
It's not just that the brain changes when it learns fear, it's that this exact microscopic addition of receptors is the fear.
Right. It's the physical embodiment of it.
All right, this is dense. Fascinating stuff. Let's do our next recap bullet summary to lock this visual in for you. Bullet 1. We know the VCA1 to BLA pathway is the specific highway for updating fear.
Yep.
Bullet 2. The mechanism is at the synapse. The brain updates fear by physically dragging extra AMPA receptors to the surface, adding more speakers between the memory cell and the fear cell. And bullet 3. Artificially removing those speakers, using 1 Hz light flashes, completely erases the avoidance behavior.
Perfectly summarized.
So the fear center is sounding the alarm. Speakers are blasting, but there's one missing link. An emotion isn't an action. How does the mouse actually move its legs to run away?
Well, that requires us to follow the domino effect to the final stage of the circuit. The amygdala, the fear center, it doesn't control the muscles. It has to send its loud alarm signal downstream to the nucleus accumbens, the NANAC, to actually coordinate the physical avoidance behavior.
The action center we mentioned earlier. And the researchers looked at 2 specific types of cells in this action center. Cells that express dopamine receptor D1 and those that express dopamine receptor D2.
And this is where the precision of how the brain processes information really shines. To figure out which cells were driving the running away, they used microendoscopic calcium imaging.
What does that mean exactly?
They implanted a microscopic lens directly into the brain of a freely walking mouse. The neurons were genetically engineered to glow bright green with a fluorescent sensor every time calcium rushed in during a firing event.
I'm kidding.
No, they could literally look at a screen and watch the cells flash like stars in the night sky in real time.
I can't even fathom the technology required to watch a single cell glow in a moving animal. And what did the starry sky show them?
It showed that only the D2 receptor cells changed their behavior after the defeat. The D1 cells didn't seem to care at all. But the way the D2 cells changed is a fundamentally different algorithm from how the fear center operates. It's the difference between rate coding and population coding.
Yes, I wanted to get into this because the logic shift here is so cool. Let me see if I have this right. Up in the fear center, the BLA, they use rate coding. After the defeat, when the mouse sees the aggressor, the exact same number of fear cells fire as before, they just fire much harder. The rate of firing goes way up because of all those extra AMPA speakers.
That's entirely correct. The individual cells work harder. But downstream, in the NAC action center, the brain switches to population coding.
Meaning more cells.
Exactly. It's not just the same cells firing faster. The signal is actively amplified by recruiting a larger crowd. More individual D2 cells suddenly join in to respond to the aggressor, and crucially, they begin to fire synchronously as a massive, coordinated group.
So, to use an analogy, the fear center is like a single alarm bell in the town square. After a trauma, the bell doesn't multiply, it just rings much, much louder. But the action center is the town itself. When that louder bell rings, the whole town wakes up.
Right. More people rush into the streets, they coordinate their movements, and they execute a synchronized evacuation.
That is amazing.
And to prove that the upstream alarm bell is what actually causes the town to evacuate, the researchers went back to their chemogenetic tools, the designer drugs. They temporarily silenced the fear center using a drug while continuing to record the glowing cells in the action center.
Let me guess. If you muffle the alarm bell, the town never wakes up.
Spot on. When the BLA was blocked, that coordinated synchronous firing in the ANAC-D2 cells never happened. The population didn't expand, they didn't synchronize, and the mouse just walked right up to the aggressor, completely failing to avoid them.
The domino effect was stopped dead in its tracks.
Okay, let's tie this whole magnificent biological machine together. This is the entire circuit, end to end. We have the identity of a specific person stored in the hippocampus, the memory center.
Yep.
When an interaction with that person turns traumatic, the synapse from the memory center to the amygdala fear center physically strengthens, the calcium rushes in, the AMPA speakers are added to the surface.
That louder signal from the fear center then travels down to the action center, recruiting a massive synchronized population of D2 cells that literally drive your muscles to walk the other way.
It is an incredibly elegant multi-stage system. It beautifully illustrates how organisms can maintain a stable, persistent memory of someone's identity. You never forget who they are, while maintaining total rapid flexibility in updating whether that person is currently a friend or a threat.
The identity file remains stable, but the downstream warning label is highly dynamic.
Right, it physically remodels itself.
Which brings us to the ultimate question. Yeah. Why should you, our listener, care about the microscopic social lives of mice? What does this mean for us, sitting in our offices or coffee shops?
Well, it provides a profound, concrete biological context to human emotional resilience. We all experience shifting social dynamics, conflicts, betrayals, and sometimes trauma. This circuitry shows us exactly how our past memories protect us.
Protect us from generalizing, you're right.
Yes. By having a stable memory network, our brains can isolate negative experiences to specific individuals, rather than generalizing that trauma to the entire world and paralyzing us with anxiety. It proves that when a relationship sours, it's not just all in your head in some abstract way. It is a literal physical change in your biology. Your synapses actually remodel themselves.
It's structural. The architecture of your brain changes based on who treats you well and who doesn't. Which leaves me with a final, somewhat provocative thought for you to mull over as we wrap up today's deep dives.
I love a good thought experiment.
We just saw that scientists can use targeted flashes of light to literally swallow up those extra AMPA receptors, right? Mm-hmm. Turning down the volume on a very specific social fear in mice, completely eliminating the avoidance behavior without erasing the memories of the individual.
Yeah, the optical LTD.
So could we one day develop therapies that selectively turn down the volume on specific human traumas or severe phobias? Imagine a medical frontier where we can leave a painful memory intact so you don't lose your history or the lessons you've learned, but we can surgically strip away the debilitating, paralyzing fear that comes with it.
That would change everything.
That's the future of psychiatry.
Mm-hmm.
It's definitely something to think about the next time you interact with that unpredictable coworker.